Hello, this is the work Analysis of Genomic Studies modeled by Citation networks, made by Paula, Jeniffer dos Santos Viriato and Jader Martins Camboim de Sá. The GWAS, Genome-Wide Association Studies, is an attempt to measure and analyze DNA sequence variations in the human genome. Efforts to identify genetic risk factors for common diseases are made in the GWAS study. It tries to make predictions about who is at risk given a certain disease. It can identify the biological foundations of disease susceptibility and develop new prevention and treatment strategies. The GWAS catalog contains a collection of genomic scale association studies. It is a collaboration with NHGRI and EMBLEBI, and it permits specialized curation and quality control. The GWAS catalog data is difficult to visualize. The GWAS catalog data has published genetic association studies versus the population that is being studied. The GWAS catalog and its links can be seen as networks and modeled like graphs. Looking at it through networks can provide implied information, such as explanation of origin and reasons for such problems and prediction of internal events through topological analysis. The objectives of this work are the creation of a network that links studies contained in the GWAS catalog with the respective populations studied, and supply other information through a network-based representation, such as related genes and phenotypes, and also given a statistical and topological study regarding the produced network. For that objective, we started data pre-processing by writing a Python code and making a mapping for each respective continent. We find matching patterns for each initial sample size and replication sample size fields, and then we create a new table listing studies and identified populations. We then go through the creation of a network of associations using the networks library, and we supply the GWAS catalog pre-processed data as input. These are the preliminary results showing the evolution from 2017 to 2020, where we can show that the correlations between studies are much more visible in the 2020 graph. Shows a big evolution in the graph representing the GWAS catalog. So, the results of this work. The data pre-processing is completed, and we obtain an archive with relationship between studies and populations. We can see a first visualization of the network, and we have a time comparison of content in the GWAS catalog. We can visualize the evolution of the association network between 2017 and 2021, as we showed before. These are the Ungroup distribution graphs, where we can see in the left here, we have on the left the different populations and on the right the different diseases, still not grouped by different populations. And we can see already that some populations are much more studied than others. So the grouping strategy is that we map each community to its respective continent, and if a community gene could express in 2 or more continents, we would replicate them for each continent. So these are the results grouped by continent, where you can see that, for example, Europe and Asia are much more studied than the other continents. Here we have some statistics showing the average path length, the nodes, the links, the average degree correlation. Here you can see the robustness of the network, where we show that by removing n nodes, how many components are lost. And we can see clearly that not many components are lost by removing endnotes, which indicates that it is a robust network. So these are the results of the analysis by continent, where you can see now in numbers that Europe and Asia are much more studied than the rest of the continents. And it shows that these studies are not proportional to the population. So we have now a few hypotheses. Hypothesis number one is that certain populations are proportionally more studied genomically than others. Hypothesis number two is that the density of studies in various diseases would offset the fact that some populations are less studied. Less studied populations would also benefit from studies carried out in other populations. What if the studied network were random? All populations would be genomically studied with the same intensity. But it is not interesting that random diseases are studied in populations. For example, recurrent diseases may not be studied in a population, and non-incident diseases can be studied but not make great advances. It is appropriate that only recurrent diseases in a population be studied. It is important that populations are studied with the same intensity, but taking into account the diseases that affect them. And all populations would benefit greatly if they were studied with the same intensity, because the studied network would become more robust, and we would know more about diseases in general. And what is observed with the results? The populations being studied unevenly is confirmed by looking at the unevenness of the studies is confirmed by looking at the number of studies for each continent. So this strengthens hypothesis number one. And despite the inequality, the network remains robust. By removing a knot, virtually no other knots are lost. So the network is robust. So the network maintains itself even while losing a few nodes. The network is critical. Each lost node represents the loss of information about diseases that affect thousands of people. It is important to remember that this network is about health, and health is a very critical area. In conclusion, it is appropriate that all populations be genomically well studied, and genomic scale studies affect knowledge of diseases such as COVID-19. We would be better prepared for the Omicron variant if we studied more African populations, for example. Thank you for your time.